Okay, welcome everyone. So it's a great pleasure to introduce Daniel Busiello, who is here as a TSVP visitor for several months, actually, until May. So Daniel earned his PhD in physics from the University of Padua in 2018, and then later was a postdoctoral fellow in the Statistical Biophysics Lab at APFL in Lausanne, and later got the prestigious Distinguished Postdoctoral Fellowship from the Max Planck Institute for Complex Systems in Dresden, and is now back in Padua as an independent group leader. Daniel's research crosses the lines between different fields. He works at the interface of information theory, non-equilibrium thermodynamics, and chemical reaction networks. And despite he's in early career science, he's already got several awards, including the Early Career Scientist Prize from UPAP in statistical physics, and last year the Ig Nobel Prize for physics for his study on the behavior of cacio e pepe pasta. So this will be the topic of another event if we manage to organize it. So Daniel, we are very happy to have you here at OIST, and the floor is yours.
So thanks for the invitation. As you can imagine, I'm not about pasta, as Simone said, so I know that probably you are hoping for something funnier than this. I try to be to reduce all technical parts to the bare minimum. In case I failed in some points and becomes too technical, stop me anytime and ask. It should be technical. Let me start from the general question that I would like to answer in this talk. So, yeah. All these things that I'm going to present here, basically they start with an idea, which is very general. And the idea is that biological systems process information from their surroundings, and they do so through several interconnected dynamical processes. And these processes can happen at very different spatial and temporal scales. So just to give you some example of why this statement is very general, the first example that I want to show you is this one. So this is a molecular transporter. I'm not going to the details of this model, but anyway, it's just a protein that imports nutrients from the external to the internal part of a cell, and it sits on a membrane, or it can also export waste from the internal to the external part of a cell. So these kind of transporters work with using several different reactions with several different rates, and in this way it can actually be shown that this system really processes the information. Then I'll explain how this processing information is defined. Process information from the outside of the cell to act in a certain way. So this is not, of course, the only example. There are also many other examples of systems that process information in very general terms. So these are neurons, of course. Also in this case, you have a lot of rates involved in neural activity, a lot of frequencies, and you also have clearly information processing in the usual sense. And we also have bacteria. So bacteria as well, they read the variation in the local nutrient concentrations. And according to that, they modulate the flagellate motion, and they do so, again, tuning a set of reactions that happen inside them. So all these systems, they all process information using several different processes, but this statement is even more true if we consider multi-component systems. One case is signaling network. This is composed by several chemical species or several different chemical processes with different rates that basically take a signal and produce a certain output. In this sense, they process some kind of information. Or even ecological networks, where we have the timescale of the bacteria and also the timescale of the whole population, gene network, and also neural networks. It's basically a collection of neurons, but we can have different regions spiking at different frequencies. And in this case, this region can also be spatially delocalized. So we can have all these processes, and of course, the establishing an accurate processing in all this system is crucial to have robust functioning of all the examples that I just shown. And usually this accurate processing is achieved through a set of complex dynamical operations and also through energy consumption. So the system consumes energy to process information, and they do so across this set of temporal and spatial scales. Just to give you an idea of how this information, how this energy is used to process information, let me go back to the first example that I show you, the one of these transporters. And I want to go into the details of the system, just to show you that this protein can be mapped into a chemical network, and basically it can be shown that transport is effective across the membrane only if there is energy available from ATP. So if the system has energy from ATP, it can actually transport something effectively, can displace the concentration inside or outside the cell of nutrients, waste, molecules, whatever. So transport can be effective in this way. This is, of course, not the only example. There are several other systems that also consume energy to work somehow. I just listed here 2 of them. Not important to go through the details. It's just to show that there is a wide variety of systems that actually fall into this category of processing information using energy. This is the case of chaperones. If you are familiar with chaperones, these are proteins that fold other proteins. And this folding process is also driven by ATP. And this is the case of photoacids. These are acids that can change the pH of a buffer solution. And they do so using energy from external light. So it's just a huge variety of systems that actually do this kind of stuff. And this energy consumption usually manifests into the presence of some fluxes in the system that break some kind of symmetry. And the symmetry that hold the equilibrium, and they are broken when we use energy. One of the symmetry might be, for example, the balance between inside and outside concentration here. So we break this symmetry by using energy and by having a net flux of importing or exporting. So we actually, with this very general picture, we actually face multiple problems. So one, I'm trying to address 2 of them. So the first one is actually, what is the role of these timescales? So I told you that there are a lot of timescales, a lot of different timescales involved in this information processing, but is that a requirement? Is actually needed to have all these timescales, and how do they affect information processing in these kind of systems? So this is the first question I would like to answer. And to give you an idea of just a proof of concept of why timescales can be important to process information, let me start with this very stupid example. This is actually a toy model. This can be, you can imagine that these are 2 molecules. 2 molecules, in each of them can be in 2 states, A1 and B1 for molecule 1, or A2 and B2 for molecule 2. And the typical timescale of this transition is tau 1. Typical timescale of this transition is tau 2. So we have 2 timescales, 2 molecules, 2 states for each molecule. Very simple model. And we also have a coupling between these two processes, these two switching processes, and these are catalytic coupling. So when molecule 1 is in the state B1, then molecule 1 can accelerate the transition of molecule 2 from A2 to B2. So don't take these too seriously. It's not really a description of molecules. It's just a model to show you that timescales are important. But anyway, there is this catalytic coupling between 2 systems. And what I can do, I can compute the modal information between layers. I'm going to use layers and processes kind of interchangeably, but when I say layers, it's just between this process and this process. Okay, so these are 2. If you look this as a network, that's basically 2 layers, each one describing a process. So what I want to study is the information that these 2 processes share, given their dynamics and this coupling. So modal information, if you're not familiar with this quantity, is basically a generalization of correlation, if you want. So it captures linear and nonlinear correlations. And this is 0 only when the joint distribution of these 2 processes is equal to the product of the marginals. So it's 0 only if these 2 processes are really independent one from each other. Okay? This is the only case in which we have 0 modal information. Otherwise, you always have something which is positive. Okay, so let's compute this modal information. So one might expect that since they are coupled, they should they should share some information, right? Which is actually true, but not always. And here the timescale actually enter the game, and we can actually study the mutual information changing the relative timescales of the two processes, so tau1 and tau2. So let's take, for example, this case, the first limiting case, where the process 1 is much faster than the process 2. Okay, so the coupling goes in this way, so 1 influences 2, but 1 is much faster, so which means that the process 2 is only feeling a sort of average of the dynamics for process 1, and this average effect will result in zero mutual information between these two processes. Even if you have a coupling, they are dynamically coupled, and if you look at the dynamics, the dynamic will be different if there is or there is no C, of course. But despite this difference in the dynamics, still these two processes have zero mutual information, exactly zero only when the timescale separation is infinitely large, of course, otherwise it's very close to zero, when process 1 is much faster than process 2. In the other limit, when process 2 is much faster than process 1, so 1 is lower, what happens is that now process 2 is feeling the instantaneous value of molecule 1, and this allows the system to actually exhibit a statistical coupling, and so a non-zero mutual information. So what I'm saying is that basically the distribution of the whole system is actually different from the case in which these two are decoupled, completely decoupled, only in this limit. So only in this limit we have a non-zero mutual information. So this is just a proof of concept to show you that the relative timescales of two processes can actually affect the mutual information, and now information propagate, yes, in the system.
What are you using for the actual variables of the process? What goes into the mutual information calculation?
This is basically a master equation process here, master equation process here. So to calculate the mutual information, basically computed the whole matrix of the whole set of states of the system. So it's basically a bipartite network that maps into a whole general master equation, and then you can do the mutual information, basically partitioning properly into the states that have molecule 1 in state A1, molecule 1 in state B1, and the other states of molecule 2. Anyway, this is not really, it's actually much more general, this statement. It's not really restricted to the fact that they are a master equation system. If you have a Langevin dynamics or multiple Langevin here, or coupled to multiple Langevin here, and the coupling is the standard coupling in Langevin dynamics, so it's a dynamical coupling. This still holds, so you can still play with timescale, and you get the same, exactly the same result. Actually, what I'm going to show you here, I'm not going to show you the dynamics that I'm going to use, at least in the first part of the talk, but the dynamics can be very general. It can be master equation, can be Langevin dynamics, can be anything. So all these statements can be proven in any dynamics. But in this case, it's
Sorry, could you make sure if your body mic is on?
Oh. Now it's on.
Maybe I can ask another question.
Yes.
What happens if I guess here in this example, coupling is directional, so it goes from 1 to 2. What happens? Does it change if it's bidirectional?
It does change. So in this case, if I also take a coupling from 1 to 2 and also from 2 to 1, meaning that, for example, when molecule 1 is in B1, it accelerates this reaction. When molecule 2 is in B2, it can accelerate one reaction here. Then in this case, you have one coupling in this direction, the other in this other direction. And in this case, you always have something which is non-zero. Because, I mean, you always have one slow process that influences a fast process. But I mean, this is just one example. Then I'm going to generalize this to every possible coupling. And this is actually the case. So let me take actually this statement that timescale influences information propagation to a more general level. So now instead of having just two processes, I have a set of processes. In this case, they are represented as discrete processes. This is not really crucial, but let's take this representation just for clarity. So these are processes where a molecule can jump between different states. Here, another molecule can jump in different states here, and so on. And this molecule can be coupled, these processes can be coupled together in a sense that the state of this process can modify the dynamics and the rates of this process. This is a usual coupling when you have distinct processes influencing each other. Okay, then we can discuss about how we can put this coupling in general context, but this is pretty natural way if you want two different processes that are coupled one to each other. And actually this, technically speaking, this mapping, this representation naturally maps into a standard coupling in a Langevin or differential equation dynamics. So it's same stuff. Okay, so these states can be anything, of course. They can be molecules that change state in a chemical network. They can be different conformational states of a protein. They can be mRNA abundances. Yes?
I just wanted to confirm that different layers are distinct processes, right?
Yes.
Like, is it possible to achieve like one layer as a consequence of coarse graining of like the faster layer, for example? Like, does it still work in that case?
Well, in continuous system, so if I describe, or if I have a set of Langevin equations here, a set of Langevin equations here, and they are coupled in the usual way, then the trivial answer is yes. So yes, I can do coarse grain. I can identify sub-layers with different timescales. In the case of a master equation, is much trickier, since the structure here, mathematically speaking, is a bipartite network. So if you have a whole network, you can never reach a bipartite network unless you really identify what you mean by bipartite, but this is something that you can do a posteriori. And the reason why it's so in master equation is that for master equation, there is one huge ambiguity in this kind of description, is that imagine that now the particle can jump from this state to this state. This is still a coupling, right? But it's a different coupling. But if you can jump from this state to this state, you basically are saying that these two processes are not really distinct processes. These are the same process in the same network. And so this is a different level of description. And indeed, this kind of jumps, they cannot be mapped into a standard coupling in a Langevin dynamics. And you can, with this, you can see that actually it's a different kind of coupling. So in principle, can be done, but it's much trickier and not always clear what does it mean to coarse grain a master equation into a bipartite network. So going on, of course, these processes can be coupled, and also the coupling at this level, they are very general. So I'm not specifying anything about the dynamics. So this can be chemical feedback modulation. In the case of neural network, they can be plasticity regulation. They also act in a similar way. I'm going to show you an example of that later. They can be epigenetic modulation. They can be anything. So it's not specifying the dynamics here. And now, from now on, I'm going to represent this set of systems in this way, also forgetting about the fact that they are discrete processes. So now I have n layers connected somehow by these couplings. And I can identify two main structures of this system. One is the causal structure, let's say. So I'm going to use the word causal, but I mean, don't be so picky about it. So I mean, it's just a word that identifies that this structure is the one constructed by the set of real couplings that I really have in the system, right? So I have a set of couplings. They define the causal structure of my network, of my whole system. But I also have another structure superimposed to this one, which is the functional structure. So since we know already that timescale or relative timescales affect information propagation, this means that maybe some of this coupling might result in a non-zero mutual information. Some other of this coupling might result in a zero mutual information, depending on the relative timescales of the processes involved. Exactly what I showed before, basically. So this means that in principle, the mutual information will not be captured, will not be present due to all these couplings. So a functional structure might be different, one that emerged from this kind of system. And in general, is different. So now the question is, can we actually say something about the functional structure just starting from the causal structure? Say that I give you n different processes, and I give you how they are coupled without specifying the dynamics. Can you say something about which coupling will actually propagate information and generate information, and which of these couplings are completely immaterial from an information perspective. Of course, the question can be also reversed, right? So if I have a set of measurement in terms of information theory, can I reconstruct part of the original couplings? The answer to the second part of the question is yes, but it's not in this talk, since we are working on right now on this project. But the answer is generally yes, you can reconstruct something, but with a slightly different measures. In this talk, I'm going to focus on the first direction. So given a causal structure, can I say something on the functional structure without specifying the dynamics? The answer, of course, is yes. So that's why I tell you this story. And basically, the answer summarizes in three main, let's say, rules or principles of this information propagation. And let me show you these rules with very simple models. I think it's actually easier than in general. So let's take this system, three processes coupled together in this way. So this is the fastest, this is intermediate, this is the slowest. So slow to fast, and then intermediate to slow. So this is a set of couplings that I have in the system. So now let's solve the system in timescale separation. I'm not going to really solve it, but I just derived the structure or the probability of the system. It is pretty intuitive to build it, actually. It can be proven that the structure is this one, but I mean, let me try to build it kind of naively. So let's start from the fast process, right? This is a process that will relax faster at steady state. This is the fastest one at play. But this process is affected by the state of process 3. So it reaches a steady state for every instantaneous value of the process 3 that is influencing its dynamics, right? So its steady state will have a conditional structure because of this slow to fast coupling. So this is P1 given the instantaneous state of 3. Then I solve the second one, P2, which is this is independent of anything. It does not have any coupling, any entering coupling. And then I solve the last one, but the last one is only affected by this intermediate layer, which is faster. And we know already that when something is faster, the only effect is an average effect. And this average effect does not really introduce any conditional dependency, since it only changes a little bit the operator, but does not include any conditional dependencies. So this is the structure that we have in this system. And from this structure only, we can actually obtain something that we call the mutual information matrix or multiscale observables, or MIMO, which just for Italians here in the room, we are not so many. This is a funny Italian nickname. I mean, so it's not MIMO, as somebody actually said, but it's MIMO, really. So we compute this MIMO here, and we see that this matrix has the only non-zero element at the point 1, 3, in the position 1, 3. Which means that the only two processes that share some information are 1 and 3, and 1 and 2 are completely disconnected, as you can imagine, but also 2 and 3 are completely disconnected. So this also comes back a little bit to the things that I just showed you in the beginning. So basically, direct interaction from fast to slow, they do not generate information. So if you have something fast influencing something slow, and you want to calculate the mutual information between these two processes, you get 0. This is, of course, in the limit of infinite separation, but I mean, if you are not infinite in separation, still it kind of works, so it's very close to 0, numerically. While feedback interaction, so from slow to fast, as we already saw, they generate information. So you have a non-zero information when something slow is affecting something fast. This also comes back to your question. When you have both of them, of course, you have always one that gives you a non-zero contribution, the other one that gives you 0. So you in general have something which is non-zero if you have both direct and feedback between the same nodes.
There's one thing that I'm not sure, because let's say in the example B, right? So if I now add an arrow from 1 to 2.
No, this is only the representation of this in terms of the functional structure. Okay. It's not another.
Let's say like if I consider another example, if you have an arrow from 3 to 1 and then from 1 to 2, then that is not something I can neglect. Okay.
So this is the third principle. The first two principles were in the first slide. The third one is in this slide, which basically now let me move the coupling from here to here, right? So it's slightly different, but this changes the structure of the probability. I'm not going again into how I build this, but it can be built in the same way. But now I have an additional conditional dependency, mathematically speaking. And then what happens is that now the mutual information matrix is non-zero everywhere. So now 3 and 1, they share information. 1 and 2, they share information. And 2 and 3, they also share information. So why? So the reason is that basically the bottom line is that information that is created by one of these feedback link, we call that feedback, but this is a slow to fast link, can be propagated downstream by direct links. So the role of direct links from fast to slow is not to generate information by themselves, but it's actually to propagate downstream something that has been generated somewhere else by a slow to fast coupling. Okay, so this is they are two very different roles. Feedback really generate information, direct only propagate information downstream. Downstream in terms of the time scales. So also this directionality here is induced by the timescale separation. It's not really intrinsic. Mutual information is symmetric, it's a symmetric quantity, so it does not have encoded any directionality. But here directionality comes only from the fact that we have a timescale ordering in the system. Okay, so we kind of answered the first question. So we understood more or less how timescale ordering affect information propagation in the system, and and of course this is relevant when we have a process that spans several timescales. So now I'd like to give you a quick example of one system we studied. We studied some of them, but this is just one case in which this kind of setting, so the presence of a dynamical process in which of several dynamical processes connected across multiple timescales, and the fact that we can identify functional structure, they have a role in understanding what the system is doing or the system can do in a certain condition. And this case is the case of neural population. So consider two neural populations, one excitatory, one inhibitory. So they are connected. These are coarse-grained neural populations, and they are connected by a set of internal interactions. They are in general nonlinear. Well, okay. And also, we consider the presence of an external input driving the system. This input can be dynamical, so can switch between different states. And this input is affecting both populations in a certain way. In this case, it's only affecting excitatory neurons, but it can affect both of them or each of them in a different way. It doesn't really change much. Now we have in the system two timescales, which is one is timescale of the input, and the other is the timescale of the internal activity of the populations. And what we see, once again, is that mutual information is non-zero only when the input is much slower than the dynamics. It's the only case in which we can see that the internal population is encoding some information on the variability of the input. Is the only way in which you can do this is just by putting an input which is slower than the intrinsic timescale of the dynamics. Otherwise, the mutual information is zero or very small. Okay, so we can actually do a little bit more in this case, not necessarily related to this idea of timescale separation. We can actually study a little bit the set of parameters in which this happens. We also see that very close to the regime of instability, I show these two together, very close to the regime of linear instability, this mutual information is maximized. So we need to have basically a slow input with respect to the timescale of internal population, and we also need to have a system which is very close to the regime of linear instability to have a maximal encoding, an optimal encoding of the information from the external input into the internal activity. Okay, the reason why you see this region here and then you see this drop is that here we really employed the usual nonlinear dynamics that is employed in the system, and this is just a linear case that shows the unstable line basically. Okay, well, but actually, what I want to do in this case is actually go into the direction of adding more timescale and see what I can say about the functional structure of the system, right? So this is the whole point of showing this. So let me add another timescale. This is the timescale of the plasticity. And plasticity is something that changes this interaction between neural population. So we have this neural population that can interact, and this interaction can also change dynamically according to some prefactor. This is a very easy way to put plasticity in place. So there are other ways. Plasticity also has its own dynamics that is affected, coupled to the dynamics of the population. And then we have this coupled system of a set of neurons connected to each other with their dynamics that is affected by the plasticity. And we also have an external input that arrives to the system. Now, 3 timescales basically: plasticity, neural activity, and external input, right? So 3 is already a good number to play a little bit with timescale and see what can happen and which kind of feature we can actually see depending on the relative timescales between these processes. I would actually like to focus on 2 limits: the limit in which plasticity is fast and the limit in which plasticity is slow with respect to the input. So we know that input has to be slower than the internal dynamics. This is a way in which these dynamics can actually encode some information. Fine. Then we also have another degree of freedom, the plasticity. And the first case is the case in which plasticity is actually slower than the input dynamics. So we have a very slow modulation of the interaction between neurons, at least much slower than the input affecting the system. It's basically a very long-term plasticity. And the question is, what is this plasticity doing? So what is the role of this kind of mechanism? So in this case, since this is the slowest degree of freedom, it basically can actually respond to a sort of average statistic of the input. So the system is affected by a variable and changing input. The plasticity in this context basically integrates the whole statistic of the input and can tune the system to a global optimum, where global means with respect to the whole statistic of the entire input. Okay, I'm not going to go into details, but basically this can be shown again with the same techniques and using the composition of probability distribution. But naively speaking, when something is slow, it's affected by an average of all the rest, which is faster. And so this is actually what plasticity is doing. So it can drive the system to a global optimum, and the global optimum coincides in some regions with an anti-Hebbian rule, in some other region with an Hebbian rule. So without going to details of what Hebbian anti-Hebbian means, in this case it just means suppression or increase of coupling strength. But it's just showing that in principle, this Hebbian and anti-Hebbian rule can actually be both optimal, depending on a specific condition in which a system are trying to encode the external input, and plasticity can actually drive the system to a global optimal point. There is also another case, as I told you already. So there is a case in which plasticity is not now slower than the input, but is here. So input is again slower than the population. We have to do that, otherwise there is no encoding at all. And plasticity now is in intermediate time scale, right? So this is not really a short-term plasticity, but it's shortish because short-term plasticity can have a different physical effect, physical weight. Yes.
So phenomenologically, where do you think the plasticity is coming from?
Phenomenologically or biologically speaking?
Well, I guess for me, phenomenon is biology, right? So, is it just intrinsic to the system, you think, or
there are several ways in which plasticity can be implemented biologically speaking, and that's why I'm a little bit careful in discussing long-term and short-term, since there might be different mechanisms controlling long-term and short-term plasticity. This plasticity we encoded, it's usually a long-term plasticity that comes from a sort of reinforcement of this connection given some repeated stimulation. So it's biologically speaking, it's a sort of internal regulation of this kind of this population. Can come from other population of neurons sending signal to the region we are looking at, for example. There are several mechanisms. So this is a sort of a phenomenological way to encode this. So basically, you are saying that the more you fire, the more you are active, the more you increase your connection or decrease your connection, depending if you are Hebbian or anti-Hebbian. So basically, the mechanism here, phenomenologically speaking, is basically a sort of self-reinforcement. But biologically, it's a different story. They can come from very different factors. And depending on the factor, you also actually have to be modeled in different terms. This is one is the simplest case.
I guess the reason I ask is because some plasticity change can happen because of neuromodulators, which themselves release because of the input. So you can imagine having maybe like a third or fourth case where like the input is also affecting the plasticity rule. Have you thought about something like that? Like
well, we did not. Fair to be honest, we did not put the input on the plasticity. It can be done for sure. The picture, I don't know exactly how the picture will change, so I need to do calculations, so I'm not entirely clear. Probably in this case, still it can act as a global optimum, since it anyway integrates the statistic of the system and the input. But it integrates also the input directly, but anyway, I think the reason why it drives to global optimum is only due to this timescale ordering. And in the same way, I also think that here it might act to perform sequence discrimination because of the same reason. So all the things that I'm going to show you here, they're not really connected to the specific dynamics, really. But of course, when also the input affects plasticity, a lot can change in the system, especially regarding the multi-stability that we see. So this can change, but in principle can be done where we didn't consider. Thanks. Okay, so just to go quickly on this part. So basically what happens here is that the plasticity now is feeling basically the instantaneous value of the input. So for every value of the input, I have a different statistics of the population and a different plasticity, right? So now I can actually select my inputs. So the set of inputs that I'm giving to the system sequentially. And let's say that I'm giving to the system these three inputs, right? A weak, an intermediate, and a strong input, right? So for each of these inputs, I have a different response of the system, a different activity, and a different plasticity that is now cannot integrate the whole statistic of the input, since now this is here. So it only see the instantaneous input arriving at the system. So one of these response can be, and in this case is, in the bistable region. What happens, of course, is that when I give this set of inputs in this order, I have a certain response of the system. This is a response in the plasticity, but I can also look at population. It doesn't change much. But when I change the order of the input, what happens is that because of this multi-stable region, also the point explored by the plasticity change. So it's not really important the order. The order is, of course, respects the order of the input, but the whole point is that this value here is different from this value here. Because actually we are passing through a multi-stable region, basically. So we are falling into another attractor, effectively, just due to the order of the inputs. So what the system is basically doing is that, and I mean, this is just a way to explore this more systematically, but anyway, what the system is doing is that basically it's performing discrimination of the order of the inputs without any fine-tuning on parameter. And this kind of discrimination is possible only due to the fact that plasticity is acting at an intermediate time scale. So this is just to say that there are ways where this timescale ordering affect the functionality of the system. And in this case, functionality is that in one case we have global optimum, in the other case we have sequence discrimination. So two very different functions that only depend on where I put plasticity in terms of timescales, not really in terms of the dynamics. Okay, how much time do you have? 10 minutes?
You don't have so much time? Yeah, total 20 minutes, should be around 10 minutes for Q&A.
Okay, okay, cool. Okay, so I can pass to the last part, which is actually pretty quick, which is basically the second question that I would like to answer in this talk, which is what is the role of energy now? So we discussed about timescale, information, but I just say that all the systems, they all require energy to work. and where is the role of energy here. So instead of giving to you details about one specific system which we can identify the energy consumption and all this stuff, I would like to discuss a slightly different problem, which is involves also the use of energy, and also another problem that you usually encounter in this kind of system, which is the fact that the information is usually limited by observation. So if you take a biological system and you imagine a set of adaptation process, the system is sometimes adapting to something that is not directly seeing in terms of observables. So we have both hidden degrees of freedom somewhere in the system and energy consumption. So the question I'm going to ask is indeed general in this case, in this sense, and it's exactly basically summarized by this picture. So imagine that we are sitting here, the door is shut, and there's another person playing a guitar on the other side of the door, but we're only hearing the music produced by the guitar. And then we ask, what is the age of this guy? And what is his nationality, or any other thing? What does he look like? Or some other questions. Of course, we don't have so many degrees of freedom to actually answer reliably to these questions. Maybe we can hear some old Italian song and then we say, okay, maybe the guy is Italian, whatever, but we cannot really reliably answer this question. But the question actually can be framed in a biochemical context, if you want, in a simple terms at least, where we can actually resort to a model where we can try to answer this kind of question, which is a similar setting. So in this case, the person who's playing the guitar is basically an active bat, which is something that produces and they consume energy that dissipates, is an out of equilibrium environment, and this active bat is coupled to a membrane, and the membrane is basically the music, which is affected by the activity of this active bat, and it oscillates because of the coupling with the active bat. Then we, or this person, is a probe, biological or artificial, doesn't matter. Then I give you one specific example in which this exact system basically can be mapped into how a red blood cell tunes its own flickering. But at this stage, it's just a probe, okay? Biological or artificial probe, which is coupled to the membrane, but is not directly coupled to the active bat hidden inside the membrane, okay? So we have this set of couplings. So the active bat is affecting the membrane is affecting the probe. So the probe can only observe the membrane, and the membrane can only observe the bat. And this is a simple model we use. We actually also use some more refined, complicated version of it. Doesn't matter, just to show you 3 equations. So the question is, given this set of coupling, we would like to ask, is it possible for the probe to get information on the bat through the membrane? So to transduce information from a hidden degree of freedom through the accessible degree of freedom. This is the general question. And the only thing that the probe can do to do that is to tune its own coupling. There are no other parameters in which the probe can actually act. If you imagine this in terms of chemical network, this might be, and actually this can be mapped to some allosteric changes that this readout molecule can induce in some protein, and these allosteric changes can modify a receptor, and the receptor is coupled to some external chemical. So this actually can have a biological interpretation. Yes.
Just so I understand, this feels like a very different problem than what you set up with what's the age of this person playing guitar. Because in this case, it's directly coupled. It's not hidden. It's not directly coupled, but it's not hidden either. Right? The degrees of freedom actually go through the chain. And I think, you know, the much harder problem is that fair? First, just
yeah, of course. I mean, of course. So yeah, of course, the information here somehow propagates in this chain. So clearly, so of course, one of the reasons why inferring the age is much more complicated problem is that, of course, I probably need an infinite number of degrees of freedom to really describe the system. Here I only need 3. So of course, this is actually, it's a matter of dimensionality as well. So in this case, I actually can identify very well the path in which this propagates. And actually, this is very interesting. You see, actually, what I'm going to show, what I wanted to show, probably I'm not going to show it, it was this one. So now the question is, what is the strategy to get this information, tuning this coupling. And one might ask, okay, the strategy is just maximizing this information that I have, that the probe has on the target, and minimizing the energy that I need to get this information. And here, this lambda basically sets the energy budget of the system. So here, the energy plays a role in the sense that if you have a limited energy budget, which is very limited, I cannot get as much information as I want between the two. But actually, what the observation was very interesting, since now what I'm doing is basically I'm cheating. So I'm using the fact that I can define this quantity. I can define the information between the probe and the target. So in this problem, the target is not really hidden. The probe is measuring its own information with respect to the target, is maximizing it. Well, all right, like this, it's easy to optimize this problem, right? So it's easy to find an optimum. So let me go then quickly on this, since you already raised the problem. And actually, what I have to do is something slightly different. So what I have to do is to maximize the information that the probe has, not the one that the probe should have. And the information that the probe has, since the probe is only observing the membrane, is information between the probe and X, the membrane. So I'm actually maximizing something which in principle has nothing to do with information between the probe and the target. I'm maximizing something which is accessible somehow. And I also minimizing the dissipation that the probe can estimate, which is only the dissipation that comes from this part of the couplings. The information that comes from this part can never be estimated, is not really accessible to any observation. Then actually, at the end, I have a more specific problem in which this idea of observation becomes more clear, and it's actually a chemical network. But in this case, just let me speak in these very general terms. But in general, these quantities are accessible in the sense that they only need the probe and something which is directly coupled to the probe. Okay, if I do that, the black line is basically what I get from the optimal case in which I directly maximize the information between the probe and the target. And this orange or red, in this case is the case in which I'm actually maximizing this effective function. Also, I'm maximizing what is observable in the system. So what you might be see immediately is that the information that the probe is getting on the target, so the one that actually my target function is this one. So this is the information that I would like to get on the hidden degree of freedom, hidden in the sense we discussed, is actually higher, can be higher, if I maximize something which is not directly this function. I mean, this might sound counterintuitive, but this is not counterintuitive since, of course, this is higher with a certain cost, with an higher cost. So I'm actually dissipating more to get more information. So this is possible, even if I only observe a fraction of my system. I can actually perform better in terms of information and poorly in terms of energy dissipation. But the point is that this dissipation here is only the one that I observe. I cannot reduce the system to this dissipation. So this contribution here is exactly the one coming from the butt. So anyway, I cannot do better than this. And my best can be better in terms of information with respect to the optimal case. Of course, at some point, the system hits a region in which this transduction becomes inefficient. So this starts going down until a region in which basically the system decides somehow to gain no information at all. And the reason is connected to exactly to dissipation. So why you have this depletion here and then zero information, is basically the system is estimating very poorly the dissipation in the system. And this poor estimation of dissipation reflects into the fact that the probe at its best would like to work at equilibrium without dissipation. And at equilibrium, also, it doesn't get any information. So the fact that it's not really capturing the real dissipation of the system reflects exactly into this regime here, But interestingly, it also gives us this nice regime of efficient transduction. So this is just connected to the timescale. Also, timescale can, again, change a lot of the system, can change the functionality and all this stuff, but I'm not going to discuss all these points. Let me just go briefly to the example that I just mentioned before. This is one example in which this thing can be somehow realized. This is a phenomenological model. It's not a biological model. So I want to be clear, very clear about this. And it's not our model somehow. So if you have a question with this model, please ask those guys, not me. But this is just a model that we use because it's basically a model that describes phenomenologically the flickering of a red blood cell and how the flickering reflects the activity of the cytoskeleton. So the model is actually very similar to the one that I proposed in the sense that cytoskeleton only affects the internal membrane, and then there is a coupling between the internal membrane and the external membrane that produce the flickering that we can observe. And the flickering is only the activity of this external membrane, basically. So this problem is very similar to the one that I just framed. So we want to understand, actually, we'd like to understand how membrane flickering reflects the activity of cytoskeleton. So what is the information between Y and eta? But in this case, we know already how much energy the cell is dissipating, because in principle, we're able to measure it. So let me skip this part, but what I'm going to show here is basically that if we do experiments and you measure flickering and you also measure dissipation, you found that there are a set of cells that are completely passive. So they produce very low entropy, and they gain very low information between the flickering and cytoskeleton. So basically, flickering is not reflecting anything about the activity of the cytoskeleton. And in this case, the cytoskeleton is also very likely is not even active somehow, or have very small activity. And there is another set of cells that actually are active. They dissipate energy, and there is also an information shared between the external membrane and the cytoskeleton. So far, so good. What is actually was nice, and at this stage only comes as an observation, so I'm not saying that this is a claim, is that we can identify in two different experiments the cases in which this transduction is efficient and the case in which is inefficient according to our framework. So we have two different sets of experiments, two different sets of measurements. The set of experiments are basically, they differ from each other by the kind of mechanical stress that they impose on the cell to measure flickering. And this kind of mechanical stress seems to correlate with the fact that transduction is efficient or inefficient according to our framework. So one set of experiments always end up in inefficient transduction. The other set of experiments always end up in efficient transduction. And they only differ by the fact that they are applying different kinds of stresses to measure flickering. Of course, it's not enough to say that this is there is a connection between transduction efficiency and mechanical conditions, but it's actually reasonable that it should be something like that for a red blood cell that actually works for mechanical stresses. Okay, so just to give you a hint, this is really a hint, so there are two slides, about what I said before. So how this then, it's also connected to the question that you asked about this observability and all this stuff. So what I did here is that basically, okay, I use now information that can be observed and dissipation that can be measured, whatever. Can this quantity really be measured by chemical systems? So can a chemical system really measure information? How? Right? So imagine that we have a molecule that arrives to the receptor. This is the membrane that we described before. The receptor now is producing an internal molecule, and this is our readout. This is our probe in the previous case. And this readout can also trigger some allosteric changes in the membrane that change also the binding affinity of the receptor. This can be, so these are cases in which this can happen. So in this setting, what this readout is really responding to is the set of binding and unbinding operation of this receptor. That's all. It's not really measuring information, dissipation, whatever. It's not measuring forces. It's not measuring diffusion coefficient. It's not measuring anything of that. It's just counting somehow fluctuations of these molecules and number of binding and unbinding events. That's all. We actually ask ourselves, is with only this set of measurement, is it really possible to measure something which goes closer to our idea of balancing information and dissipation in a system? Well, the answer is actually yes, also to our surprise, that using counting statistics, you can actually estimate mutual information. This is pretty obvious, right? Counting statistics, basically, you can actually obtain an empirical distribution. From this empirical distribution, you can get an estimate of the information somehow, processing, of course, this information, but you don't need more than just counting events. And the other point is that, the crucial one actually, is can I estimate dissipation? Of course not. I cannot estimate dissipation directly, since dissipation is not directly a time-symmetric observable. Actually, it's time antisymmetric. So I cannot measure dissipation through counting, but what I can do is I can measure some kinetic observables through these counting statistics. But what is really nice is that if you set the balance between information and dissipation, is that this counting statistics and this kinetic information is already enough to add a very good estimation of dissipation, even better than the one that I just showed you before, estimating forces. So this actually works even better than the framework that I showed you before. This is actually working better in terms of how well the system is estimating dissipation using kinetic observables. So counting, basically counting events, no more than that. Taking derivatives, but there are just differences. Okay, so this is the end. These are just the things that I said several times during the talk. So I'll just give you on the slides these messages. And let me thank my main collaborators in this project. These are Giorgio, Giacomo, and Ivan. And thanks you for your attention.
I think we have time for maybe one or two questions.
Yeah, so for the information dissipation balance, I have a question that's on both sides. So one is, there are things that matter to a living system and things that don't, which aren't captured by just bits. I'm curious about that. There's more meaningful bits than others. And is dissipation here real dissipation? So, if you could measure real dissipation in your system, how close is your measure to something that matters for the cell? I'm just curious about those. Yeah.
So let me answer the first part, which is somehow easier. Easier to answer, not easier to really respond to the question. So the point is that we propose these kind of schemes. We propose that information and dissipation might be important to decide what the cell has to do, or how it has to change some receptor dynamics or whatever. It's not really clear if this is really important. So it's not really clear what are the relevant information that one can extrapolate.
Right, like I might spend a ton of energy to get away from a tiger. That's a very rare,
Exactly. So it's anyway, these things that have to be, let's say, verified, or at least connected to some problem, real problem, where we actually can measure these kind of things and actually understand more or less what the system is doing, and if it somehow respects the framework that we try to build. Information dissipation are two general quantities, pretty natural to discuss in terms of what the biological system is doing. So that's why we're thinking to these specific features. Of course, there may be much more than that. You can actually play with some kind of correlation instead of information. It may be already maybe enough, instead of just evaluating the whole information landscape of the system. But of course, we don't know exactly the answer to this question. That's why we studied this red blood cell, because at some point we said, okay, probably this example is already a good starting point. Of course, much more experiments are needed, much more mechanical conditions to see if they really respect what we are measuring and so on. Concerning dissipation, my answer is no. if you ask to other people, the answer may be different. My answer is no, we are not measuring the dissipation that you obtain with a calorimetric measurement on the system. In the case of red blood cell, apparently they did it. So that model with those parameters capture the whole calorimetric dissipation. This might be questionable, maybe not, whatever. They basically infer some parameters in this phenomenological model that is able to capture the calorimetric dissipation. So in this respect, that model was capturing the whole dissipation. But if you ask me if this dissipation that I capture here is the whole dissipation of the whole system, is the dissipation of this system, the one that I'm modeling, nothing else. So if there are some other processes coupled to that, if there is a dissipation coming from this internal degrees of freedom that I'm ignoring and all this kind of stuff, is there some dissipation coming from, let's say, entropic changes, I cannot capture that. But this is a known problem somehow in stochastic thermodynamics, in general in this framework. So we are aware of that. But anyway, we are confident the cell is only measuring something which is accessible, and hopefully that we can also measure a little bit. There was another question.
Daniel is here for a few months, so we are interested in talking with you. So I think
one of the main, the foundation of the whole thing that we are looking at, which is basically very rightly mentioned, is the time scale. And then from that perspective, one thing that you mentioned was if the input is going to be faster than the system that is actually observing it, no information is going to be gained. But this is only, correct me on this, I'm trying to understand, I'm not questioning. This is only if we consider the same dimensions of the input that is being received as of the system and the input. Because, like, if we have a system that observes its work through establishing the state space in which every dimension would be a derivative of the previous one, and that derivative is actually coming from the time scale of the system itself, it can approximate the input. It gets higher dimension, but so I mean, think about it like you have a very fast input coming in, right?
Mm-hmm.
And then you establish. Of course, you also mentioned that your limit is going to be infinity. We cannot get, but we can approximate it by the fact of if you increase the number of dimensions to a very large number with a very small time tau. So you're going to have a t minus tau, and every dimension is going to be.
Okay.
Right?
I see.
And in that sense, you can actually generalize in this space and approximate what you're observing. You are not running as fast as the system, as fast as the input, but you are slowing down the input to the time scale that the system can manage. So
okay, let me see if I got it correctly. So so basically there are several layers in which I can answer briefly. So when the dimensionality are not important, like roughly speaking, but if with dimensionality you also associate a certain time scale to every specific input, and you span several time scales in your input, you basically
the input we don't consider it. Like just like that Markovian chain that you showed, that you said that you cannot actually say anything about the original input. You can approximate it. We can have a lower limit on it. To some certain level, we can understand it based on the information processing inequality, right? But we can approximate. We cannot say exactly what it is, but we can approximate. But for this dimension, what I mean is the system. So the system, instead of dealing with what the input is, deals with the state space of the input.
I think in the interest of time, I'm going to close it here.
Okay, but we can
a quite specific question.
Yeah, we can discuss. Because I mean,
which is not questioning what you present.
No, don't worry. Yeah. I'm trying to understand. To frame exactly what you said in this framework, because I fear that it might just be a matter of the dynamics, not really of information. Because dynamics and information, they can stay at two very different levels. So you can have an effect on dynamics, but zero information anyway. So it might even be just that.
Okay. Let's end this together.
Thanks.